You're walking through a canyon, or simply taking a stroll on a beach, when you find something small. It's a rock, and it has something interesting hiding inside it. You think it looks like animal remains, but you're not really sure. How can an animal be stuck in a rock? What even is this rock? Could this be important for the scientific world?
Well, what you just found is simply called a fossil, and what you're looking at is a glimpse of life from more than a few million years ago. Famous biologist and paleontologist Greg Cord Gaylord Simpson once estimated that out of all the animals that have ever evolved on Earth, roughly 99% of them have gone extinct.
Stories of most of these creatures will never be known because they simply left no traces of themselves behind. But sometimes, if the circumstances are just right, their remains can be preserved in forms of fossils.
Actually, fossils are just fragments of animals, most commonly bones or teeth, that have mineralized and become stone. Soft tissue mostly doesn't get preserved, simply because there are many scavengers waiting within the food chain. So, bones and teeth, maybe even some feathers, are usually what's left behind.
Other than animal remains, plant tissue can also be fossilized. Although it is easily degraded in most cases, pollen is often well preserved because of its exine, the extremely durable protective outer layer.
The most likely way an animal is going to get fossilized is if it lived in or near some body of water. This increases the likelihood of the dead carcass being covered by mud or sand. Consequently, most fossil-bearing rocks are formed in water. You may know these rocks as sedimentary rocks. So, these are the ideal circumstances for a creature to get fossilized. But why are these specific parts of animals the ones that get preserved most often?
Bones, teeth, hard tissue in general, is made out of minerals, more specifically calcium phosphate, which can be preserved largely unchanged in structure or in composition. In the process of fossilization, remains that are covered in soil or submerged in water become further mineralized by other substances, such as silica or calcite minerals.
Through tiny pores on the animal matter, these minerals can enter and attach themselves to the cellular walls. Over time, they create crystallized networks in the empty crevices within the tissue. This way, what was once living matter turns into stone. This specific process of mineralization of tissue is called permineralization, also known as petrification.
Even if the bones have been successfully buried and petrified, doesn't guarantee that they will remain intact. The longer the fossil is buried, the greater the chance of its destruction becomes. Shifting rock layers and even glacier movements can disrupt and destroy whole sedimentary layers of the Earth's crust.
During the last ice age, the Arctic glaciers destroyed a big part of North America's sedimentary rock layers, and with them, most of the fossils that were buried there. We are also less likely to find fossils in older layers.
So although in theory you could just drill down till you get to the right depth where you can find fossils, this is rather expensive and unpractical. So paleontologists don't do this. They visit natural exposure sites where they have access to multiple sedimentary layers at once. These are places like canyons or cascading mountain walls.
Of course, other processes or methods also exist, such as carbonization, in which usually soft tissue gets replaced by a thin layer of carbon. Petrification and carbonization create a type of fossil known as altered fossils. They are called this way because organic matter undergoes physical or chemical changes.
Remains can also be preserved in tar pits, frozen in ice, covered in amber, or mummified. Fossils made this way belong to the other group, known as unaltered fossils, where organic matter and bone composition is retained. These fossils are rare, but are especially important because they show unchanged forms of an organism. They exactly preserve all the textures and shape that the animal had. The importance of this becomes clear if we look at the skeletal structures of elephants. Looking at their skull alone doesn't tell you that the animal has giant ears and a long trunk.
Same goes for their relatives, woolly mammoths. They've gone extinct around 10,000 years ago, but we still have some specimens that are fully preserved because they were frozen in Arctic areas of Siberia and Alaska. Not only do they show skeletal structure, they also preserve hair, muscle, the digestive tract, and even what they ate in their last moments of life. DNA can also be extracted from some of these specimens and can later be used in genetic analysis. Because of that, we can confidently say that woolly mammoths had at least two color variants, ones with lighter and ones with darker coats.
Similarly, scientists were able to extract enough DNA from Neanderthal skulls to isolate the gene responsible for hair color. They discovered that it's the same one that gives present-day humans red hair, so we know some Neanderthals were redheads.
Amber is a great way for smaller animals or even plants to get preserved. Insects are most commonly found in amber, but small lizards and bird feathers have also been found. Of course, this method is most suitable for smaller organisms. All of this tells us very little about the life of the animal, but certain behaviors can be preserved in forms of imprints.
If an animal lived in a swamp where soil is soft, their footsteps can be fossilized. Or if volcanic eruption happened relatively close to where the animal was, their walking path through volcanic ash can also be preserved due to rapid cooling.
Plants also leave their imprints on the area around them, most importantly with the pollen and spores they spread. Looking at the outer structure of preserved pollen grain can let scientists determine what the flora of specific area was like, since the shape of a pollen grain is very unique to a specific group of plants and adaptations they had.
Of course, there also exist fossils of dinosaurs who have died shielding their nests from volcanic ash, and even dinosaurs who were in the midst of battle when sand dunes fell on top of them and captured their fight forever.
On the topic of nests, eggs are also very important fossils. Since most dinosaurs were enormous animals, the likelihood of finding all of the bones of one animal in one place is very small. So, if the circumstances were just right, in a fossilized egg, you can see the entire baby animal and all the bones it had. This helps with identifying and differentiating different species.
Okay, now you know what a fossil is and how it's created, but how do you determine its age? And how to compare it to other specimens?
Different techniques are used, and preferably you should use more than one to verify the age of your fossil. Stratigraphy is one of the oldest methods of determining how old a fossil is. It's a method of placing fossils in relative sequence to each other.
It's a technique dating all the way back to 1760, when Giovanni Arduino decided to arrange rock layers from oldest to youngest, from those that were deepest in the ground to those that were closest to the surface.
Each layer of rock is called time horizon because it contains the remains of organisms from one slice in time. This gives no absolute age, but it provides a chronological sequence of fossil species relative to each other.
Adding to that, index fossils only appear within one specific time horizon. They represent markers that can be used to match rock layers of same origin that over time drifted apart due to tectonic movements.
Pollen is a good index fossil that plants create, and from the animal kingdom, the most often used index fossils are preserved isopods.
When dealing with plant matter, palynomorphs, the microscopic remains of plants, algae, and fungi, have an important role in biodating the stone they are found in.
One of the newer methods of dating fossils is radiometric dating. This method utilizes the natural transformation of an unstable elemental isotope to a more stable form over time.
The decay of an element from one isotope state to another occurs at a constant rate. The length of time that must pass before half of the atoms have transformed into product atoms is called half-life.
When we compare the atom ratio of product to original, and if we know the rate at which the transformation occurs, then the age of the rock and the age of the fossil it holds can be calculated.
Specifically, carbon dating is used to determine the age of organic materials. Half-life of the isotope carbon-14 is roughly 5,730 years, Carbon-14 is the most commonly found form of carbon in living organisms, while carbon-12 is most commonly found in the environment.
After the death of an organism, carbon-14 starts to decay into carbon-12. By looking at the ratio of carbon-12 to carbon-14, we can determine how old something is. One of the big limitations of carbon-metric dating is that it's considered accurate up to 50,000 years, after which the number of carbon-14 isotopes becomes too small to measure.
The potassium-argon series is another form of radiometric dating that relies on the half-life of potassium-40 to argon-40, which is 1.3 billion years. It's useful for determining how old the rocks around the fossils are, because argon is a noble gas and doesn't interact with minerals around itself. The only way for the ratio of argon to potassium to go back to zero is for the rock itself to be melted and argon to be released into the atmosphere. This is highly effective for dating volcanic rocks and minerals older than 100,000 years.
So, if we find a rock that has very little argon in it, we can estimate the age of the rock to be quite young. And if we find a rock that has lots of argon in it, we can assume it's quite old. Since the method is only used on rocks itself, the age of fossils is determined by proxy.
Fossils are one of the most valuable means for understanding the history of life on Earth. They provide direct physical records of organisms that lived millions of years ago, allowing scientists to reconstruct how species looked, lived, and evolved over time.
Through careful study, fossils reveal patterns of change, extinction, and adaptation that would otherwise remain unknown. However, this knowledge depends highly on accurate calculations based on current findings. Fossils must be analyzed in their proper geological and biological context, using established scientific methods rather than being randomly combined.
Mixing unrelated fossils to create imagined species can only lead to false conclusions about evolution. Fossil research not only helps us visualize ancient ecosystems, but also strengthens our understanding of biology, evolution, and the processes that shape life on Earth. Fossils also hold great importance as an energy resource. Petrified trees that lived in forests during the Carboniferous period, more than 350 million years ago, are the largest source of coal.
Same goes for petroleum, also known as crude oil or simply oil. It is mostly made from anaerobic decay of Mesozoic plankton and algae. Life as we know it would be impossible to imagine without things such as plastic, rubber, and diesel fuel that are direct derivatives of petroleum.
So, now you know that fossils are not just pretty rocks, but are also important economic assets and are fundamental for understanding evolution of life on Earth.